dose and contributed to the main safety data set. Among these 37,706 participants, 49% were female, 83% were white, 9% were Black or African American, 28% were Hispanic or Latinx, 35% were obese body mass index, the weight in kilograms divided by the square of the height in meters, of at least 30.0, and 21% had at least one coexisting condition. The median age was 52 years, and 42% of participants were older than 55 years of age. Table 1 and Table S2. Safety, local reactogenicity, Figure 2. Local and systemic reactions reported within 7 days after injection of BNT162b2 or placebo. According to age group, the reactogenicity subset included 8,183 participants. Overall, BNT162b2 recipients reported more local reactions than placebo recipients. Among BNT162b2 recipients, mild to moderate pain at the injection site within 7 days after an injection was the most commonly reported local reaction, with less than 1% of participants across all age groups reporting severe pain. Figure 2. Pain was reported less frequently among participants older than 55 years of age. 71% reported pain after the first dose, 66% after the second dose, than among younger participants, 83% after the first dose, 78% after the second dose. A noticeably lower percentage of participants reported injection site redness or swelling. The proportion of participants reporting local reactions did not increase after the second dose. Figure 2a. And no participant reported a grade 4 local reaction. In general, local reactions were mostly mild to moderate in severity and resolved within 1 to 2 days. Systemic reactogenicity. Systemic events were reported more often by younger vaccine recipients, 16 to 55 years of age, than by older vaccine recipients, more than 55 years of age, in the reactogenicity subset and more often after dose 2 than dose 1. Figure 2b. The most commonly reported systemic events were fatigue and headache, 59% and 52%, respectively, after the second dose, among younger vaccine recipients, 51% and 39% among older recipients, although fatigue and headache were also reported by many placebo recipients, 23% and 24%, respectively, after the second dose, among younger vaccine recipients, 17% and 14% among older recipients. The frequency of any severe systemic event after the first dose was 0.9% or less. Severe systemic events were reported in less than 2% of vaccine recipients after either dose, except for fatigue, in 3.8%, and headache, in 2.0%, after the second dose. Fever, temperature, 38 degrees Celsius, was reported after the second dose by 16% of younger vaccine recipients and by 11% of older recipients. Only 0.2% of vaccine recipients and 0.1% of placebo recipients reported fever, temperature, 38.9 to 40 degrees Celsius, after the first dose, as compared with 0.8% and 0.1%, respectively, after the second dose. 2 participants each in the vaccine and placebo groups reported temperatures above 40.0 degrees Celsius. Younger vaccine recipients were more likely to use antipyretic or pain medication, 28% after dose 1, 45% after dose 2, than older vaccine recipients, 20% after dose 1, 38% after dose 2, and placebo recipients were less likely, 10 to 14%, than vaccine recipients to use the medications, regardless of age or dose. Systemic events including fever and chills were observed with the first 1 to 2 days after vaccination and resolved shortly thereafter. Daily use of the electronic diary ranged from 90 to 93% for each day after the first dose and from 75 to 83% for each day after the second dose. No difference was noted between the BNT162b2 group and the placebo group. Adverse events. Adverse event analyses are provided for all enrolled 43,252 participants, with a variable follow-up time after dose 1, Table S3. More BNT162b2 recipients than placebo recipients reported any adverse event, 27% and 12%, respectively, or a related adverse event, 21% and 5%. This distribution largely reflects the inclusion of transient reactogenicity events, which were reported as adverse events more commonly by vaccine recipients than by placebo recipients. 64 vaccine recipients, 0.3%, and 6 placebo recipients, 0.1%, reported lymphadenopathy. Few participants in either group had severe adverse events, serious adverse events, or adverse events leading to withdrawal from the trial. 4 related serious adverse events were reported among BNT162b2 recipients: shoulder injury related to vaccine administration, right axillary lymphadenopathy, paroxysmal or ventricular arrhythmia, and right leg paresthesia. 2 BNT162b2 recipients died, 1 from arteriosclerosis, 1 from cardiac arrest, as did 4 placebo recipients, 2 from unknown causes, 1 from hemorrhagic stroke, and 1 from myocardial infarction. No deaths were considered by the investigators to be related to the vaccine or placebo. No COVID-19 associated deaths were observed. No stopping rules were met during the reporting period. Safety monitoring will continue for 2 years after administration of the second dose of vaccine. Efficacy. Table 2. Vaccine efficacy against COVID-19 at least 7 days after the second dose. Table 3. Vaccine efficacy overall and by subgroup in participants without evidence of infection before 7 days after dose 2. Figure 3. Efficacy of BNT162b2 against COVID-19 after the first dose. Among 36,523 participants who had no evidence of existing or prior SARS-CoV-2 infection. 8 cases of COVID-19 with onset at least 7 days after the second dose were observed among vaccine recipients and 162 among placebo recipients. This case split corresponds to 95.0% vaccine efficacy, 95% confidence interval, CI, 90.3 to 97.6, Table 2. Among participants within those without evidence of prior SARS-CoV-2 infection, 9 cases of COVID-19 at least 7 days after the second dose were observed among vaccine recipients and 169 among placebo recipients, corresponding to 94.6% vaccine efficacy, 95% CI, 89.9 to 97.3. Supplemental analyses indicated that vaccine efficacy among subgroups defined by age, sex, race, ethnicity, obesity, and presence of a coexisting condition was generally consistent with that observed in the overall population. Table 3 and Table S4. Vaccine efficacy among participants with hypertension was analyzed separately but was consistent with the other subgroup analyses. Vaccine efficacy, 94.6%. 95% CI, 68.7 to 99.9. Case split, BNT162b2, 2 cases, placebo, 44 cases. Figure 3. Shows cases of COVID-19 or severe COVID-19 with onset at any time after the first dose. MIT population. Additional data on severe COVID-19 are available in Table S5. Between the first dose and the second dose, 39 cases in the BNT162b2 group and 82 cases in the placebo group were observed, resulting in a vaccine efficacy of 52%, 95% CI, 29.5 to 68.4. During this interval and indicating early protection by the vaccine, starting as soon as 12 days after the first dose. Discussion: a 2-dose regimen of BNT162b2, 30 per dose, given 21 days apart, was found to be safe and 95% effective against COVID-19. The vaccine met both primary efficacy endpoints, with more than a 99.99% probability of a true vaccine efficacy greater than 30%. These results met our pre-specified success criteria, which were to establish a probability above 98.6% of true vaccine efficacy being greater than 30%, and greatly exceeded the minimum FDA criteria for authorization 9. Although the study was not powered to definitively assess efficacy by subgroup, the point estimates of efficacy for subgroups based on age, sex, race, ethnicity, body mass index, or the presence of an underlying condition associated with a high risk of COVID-19 complications are also high. For all analyzed subgroups in which more than 10 cases of COVID-19 occurred, the lower limit of the 95% confidence interval for efficacy was more than 30%. The cumulative incidence of COVID-19 cases over time among placebo and vaccine recipients begins to diverge by 12 days after the first dose, 7 days after the estimated median viral incubation period of 5 days, 10, indicating the early onset of a partially protective effect of immunization. The study was not designed to assess the efficacy of a single-dose regimen. Nevertheless, in the interval between the first and second doses, the observed vaccine efficacy against COVID-19 was 52%, and in the first 7 days after dose 2, it was 91%, reaching full efficacy against disease with onset at least 7 days after dose 2. Of the 10 cases of severe COVID-19 that were observed after the first dose, only 1 occurred in the vaccine group. This finding is consistent with overall high efficacy against all COVID-19 cases. The severe case split provides preliminary evidence of vaccine-mediated protection against severe disease, alleviating many of the theoretical concerns over vaccine-mediated disease enhancement. 11. The favorable safety profile observed during Phase 1 testing of BNT162b2 8 was confirmed in the Phase 2/3 portion of the trial. As in Phase 1, reactogenicity was generally mild or moderate, and reactions were less common and milder in older adults than in younger adults. Systemic reactogenicity was more common and severe after the second dose than after the first dose, although local reactogenicity was similar after the 2 doses. Severe fatigue was observed in approximately 4% of BNT162b2 recipients, which is higher than that observed in recipients of some vaccines recommended for older adults. 12. This rate of severe fatigue is also lower than that observed in recipients of another approved viral vaccine for older adults. 13. Overall, reactogenicity events were transient and resolved within a couple of days after onset. Lymphadenopathy, which generally resolved within 10 days, is likely to have resulted from a robust vaccine-elicited immune response. The incidence of serious adverse events was similar in the vaccine and placebo groups, 0.6% and 0.5%, respectively. This trial and its preliminary report have several limitations. With approximately 19,000 participants per group in the subset of participants with a median follow-up time of 2 months after the second dose, the study has more than 83% probability of detecting at least one adverse event. If the true incidence is 0.01%, but it is not large enough to detect less common adverse events reliably. This report includes 2 months of follow-up after the second dose of vaccine for half the trial participants and up to 14 weeks maximum follow-up for a smaller subset. Therefore, both the occurrence of adverse events more than 2 to 3.5 months after the second dose and more comprehensive information on the duration of protection remain to be determined. Although the study was designed to follow participants for safety and efficacy for 2 years after the second dose, given the high vaccine efficacy, ethical and practical barriers prevent following placebo recipients for 2 years without offering active immunization. Once the vaccine is approved by regulators and recommended by public health authorities, assessment of long-term safety and efficacy for this vaccine will occur, but it cannot be in the context of maintaining a placebo group for the planned follow-up period of 2 years after the second dose. These data do not address whether vaccination prevents a symptomatic infection, a serologic endpoint that can detect a history of infection regardless of whether symptoms were present. SARS-CoV-2 binding antibody will be reported later. Furthermore, given the high vaccine efficacy and the low number of vaccine breakthrough cases, potential establishment of a correlative protection has not been feasible at the time of this report. This report does not address the prevention of COVID-19 in other populations, such as younger adolescents, children, and pregnant women. Safety and immune response data from this trial after immunization of adolescents 12 to 15 years of age will be reported subsequently, and additional studies are planned to evaluate BNT162b2 in pregnant women, children younger than 12 years, and those in special risk groups, such as immunocompromised persons. Although the vaccine can be stored for up to 5 days at standard refrigerator temperatures once ready for use, very cold temperatures are required for shipping and longer storage. The current cold storage requirement may be alleviated by ongoing stability studies and formulation optimization, which may also be described in subsequent reports. The data presented in this report have significance beyond the performance of this vaccine candidate. The results demonstrate that COVID-19 can be prevented by immunization, provide proof of concept that RNA-based vaccines are a promising new approach for protecting humans against infectious diseases, and demonstrate the speed with which